Okay, it's now my great privilege to introduce Marna Whittington to the stage. Marna is going to moderate our panel, and Marna is a remarkable individual. She has her master's and her PhD in quantitative analysis from Pittsburgh. She has served on the board. She has served in many different companies over the years, the Global Alliances, Morgan Stanley Investment Group, and a large number of other companies doing cutting-edge analysis, which is similar but different from what we do within the Cancer Center. She has been on the board and involved with Salk since 2005. She has always been one of our more stalwart, consistent, and dependable partners. It is lucky for all of us to have Marna here, and so it's my great pleasure to have her moderate this panel. Thank you.
Thank you, Ruben, and welcome to all of you. It's so good to have you with us here tonight. My favorite activity when I come to Salk is to listen to our scientists talk about the research that is going on in their laboratories. And we have a wonderful opportunity with our panel. 33 of our 53 research labs here at Salk are involved in trying to conquer cancer. Over half our faculty are involved in this cancer initiative. Today we have 4 of our faculty with us on our panel. We have Ron Evans. You guys are eager. Diana Hargreaves. Sue Keck and Professor Jeffrey Wall. They're going to introduce themselves a little more fully in a minute, but I just wanted to tell you that our panel discussion is built around the questions that you all so enthusiastically submitted when you asked us to help, when we asked you to help frame the discussion. So, given that, let's get started. You're on.
Thank you very much, Marna. Thank you, Ruben, and thank all of you for coming today, for your interest in the Salk, and for your passion that we share about conquering cancer. I've been here for 37 years, and during all of that time, I've devoted my research to studying and conquering cancer. And the reason for this is very personal to me. My dad was a lifelong smoker, and when he died, he had a massive lung cancer. This is a cancer that could have been prevented. My mother died of colon cancer. This is a cancer that if she had agreed to have colonoscopies earlier, she could have been treated more effectively, and maybe she would have lived to see my son's bar mitzvah. So to me, it's very personal. Now, as you've heard from Ruben, the Salk Institute is a place that you go into the courtyard and the only thing that you can see is the horizon. We're challenged, all of us on this panel, all of my colleagues at the Salk, to address and answer the imagine if questions. These are the big questions that when they're answered, not if they're answered, transform the way we think about cancer and other problems. I'm going to give you an example of one of the imagine if questions that I've addressed with regards to triple-negative breast cancer, because this is a form of breast cancer for which there are no personalized therapies currently available. I have different glasses now. Now, how is it that we, no, I'm going backwards. How is it that we go about answering this question? First, let me tell you that when triple-negative breast cancers are diagnosed, they present as masses of many different kinds of cells. As you all probably know, cancers start from an initial cell. How is it that this initial cell gains the ability to shapeshift, to morph over time into many different kinds of cells that we see in the cancer at diagnosis. Answering this question is really important because it's within this diversity of cells that are within the cancer lie the roots of drug resistance and metastasis, the spread of cells to distant parts of the body that ultimately kills a person. How can we answer this question? How do these cells arise? Well, we get a clue from listening to the pathologists who have told us that cancer is a caricature of normal development. Here you see a picture of a normal mammary gland, and reflected in that picture is the cancer. The cancer is called that because of the Greek karkinos, which means crab, because the ancient physicians, when they found a cancer, would see that all of the blood vessels that fed the cancer looked like the legs of the crab. Crab, karkinos, cancer. I'll get back to the importance of that at the very end. So, the pathologists have told us that cancer is a caricature of normal development. What we've done is to look at the roots of cancer in the roots of development, looking at the development of the mammary gland starting at the very earliest times in the embryo. And we made a startling and unexpected discovery. And that is that the stem cells that generate the mammary gland in the adult, triple-negative breast cancers, seem to morph themselves into this type of very primitive cell. That is the most plastic cell in the mammary gland. It is designed to give rise to different cell types. And so what happens in cancer is they reprise an old program, the one used at the beginning of development, to gain their special powers that have evaded physicians for so long to effectively treat them. So the features that we find in these fetal mammary stem cells are now starting to give us clues as to how to treat metastatic breast cancer. Because just recently, we've discovered a previously unsuspected pathway. Again, how these cells reprise a primitive embryonic strategy that's involved in the normal movement of cells from one place to another, enabling the cells that move to start setting up shop in that new site. These are embryonic-like cells in an adult body that provides beautiful opportunities for selective therapeutic intervention. We're extremely excited about this. So let me just sum up by saying the following. In Greek mythology, it was Heracles who crushed Karkinos. Let us think of cancer research as our Heracles, because I strongly believe that it is only cancer research that will turn cancer into the four-letter word that every patient wants to hear: gone. Thank you. It is my great pleasure to introduce my colleague and neighbor with whom we're already collaborating, Dr. Sukek.
So thank you very much, and it is an extreme pleasure to be here with you and to share some of our insights into cancer and the cutting edge of what's happening right now in this field. And I'm Susan Keck. I'm the director of the NOMA Center for Immunobiology and Microbial Pathogenesis. And I'm an immunologist, so you might be wondering, what's an immunologist doing up here at a cancer panel? And that's because we now have evidence, undeniable evidence, that our immune system can be unleashed to fight cancer. And a particular cell type in our immune system called a T cell is critical to this process, and that's what I study. I'm trying to understand how T cells form, how they function, and how they help us fight infectious disease, and how they can help us fight cancer. A year ago, when I turned 45, it was a very personal time for me, because that was the age at which my mother died of breast cancer. And as painful as it was for me to watch her die, as I'm sure many of you have also experienced your loved ones, it's just a tragic, atrocious process. And as a young teenager, it really affected me, but what I saw in my mother when she was dying was extreme strength and courage, and in ways I've never seen before. And that I saw as gifts that she was giving to me for me to pursue my own dreams and my own passions and scientific research, to study and to never give up on these major challenges. Now, I haven't been studying cancer as long as my friend Jeff here, and that's in part because it's only been in the last decade that us immunologists have been empowered now to use the immune system to fight cancer. And for many years, the cancer researchers have been looking for magic bullets. What are these drugs selectively that we can find that can be used to precisely target the immune system, the cancer cells. Let's see if it's going there. These magic bullets that can precisely target the cancer cell but not hurt the healthy cells. And we know we've fallen short of this goal tremendously with the very blunt weapons that we have of chemotherapy and of radiation. Let me just run through that again. Sorry, I went too fast. Because what we know now is that these magic bullets are actually our T cells. They've been inside our bodies all this time. And now we have the power to use these T cells like magic bullets to go on and to attack and destroy the cancer, because that's what T cells were built to do. T cells are built to go and search out and destroy the unhealthy, the stress, the damage, the infected cells. And that's what they're able to do. But there's a big challenge. And the challenge is that why doesn't this work in our cancers all the time? Because T cells, when they enter the tumor, the environment is so hostile, it's so repressive to the functions, it actually turns the T cells off, and it stops them from attacking the cancer. And this is the basis of immunotherapy, a revolutionary form of medicine that probably many of you have heard about already. Maybe some of you have actually been receiving. What immunotherapy is doing for cancer is using drugs that can stimulate and re-energize our immune system, and particularly our T cells. These are one of the vital targets of immunotherapy, to re-energize them to attack and destroy the cancer and return to a healthy tissue. So imagine if, taking from Jeff, imagine if our immune system could cure cancer like it does a viral infection. And one thing that our lab is trying to understand is what are these breaks that are stopping the T cells from functioning in this tumor microenvironment? And can we use that information to then stop the T cells from listening to those breaks, having them evade those breaks, and be able to regain function? And can we design T cells that are better, more fit, able to last longer, and work harder in the tumor to destroy these tumors? So thank you very much for joining us on this pursuit. And now I'd like to introduce Diana Hargreaves.
Thank you, Sue. Yes, my name is Diana Hargreaves, and I am an assistant professor in the Laboratory of Molecular and Cell Biology here under Tony Hunter. And I've been here for just about 2.5 years. And like many of you, I have family members that have been affected by cancer. My father is undergoing treatment for prostate cancer right now. And although I'm very aware of the side effects that accompany these therapies, such as chemotherapy and radiation therapy, it's very difficult to watch a loved one go through this, and also to be aware of the sort of permanent and lasting effects that these therapies and treatments can have. And so we're all after that precision medicine that can eradicate the cancer but leave the healthy cells intact. And you heard from Sue about this really revolutionary new approach of immunotherapy. Well, we're interested in a complementary approach, which I'll call tweaking the epigenome. So the epigenome refers to this layer of chemical modifications or molecular flags that lay on top of the DNA sequence and tell the cell which genes to turn off and which genes to turn on. This is very important during normal development, and it is the basis for why, for example, your brain cells are different from your muscle cells. But it turns out that in cancer, cancer genes are flagged, and in particular, cancer-promoting genes or tumor-promoting genes are flagged. In fact, the cancer cell likes to ramp up the placement of these flags to really produce very high levels of these tumor-promoting genes. And in fact, in many cancers, the enzymes that place the molecular flags are actually mutated, leading to widespread dysregulation of the epigenome. And so this gives us really a window of opportunity for what we'd like to call drugging the epigenome. And this is the idea that we can develop drugs to block the enzymes that place these molecular flags and thereby reset the genome to its normal levels and return the cancer cell to its normal state. And these drugs have already been in development and are starting to enter clinical trials with definitely some efficacy. So our lab is particularly focused on ovarian and gynecological cancers, in particular those cancers that have mutations in these epigenetic regulators. There we really know the root of the problem. We're trying to understand how these mutations in the enzymes themselves impact the epigenome and gene expression to promote cancer development and cancer progression. And again, the idea of using these drugs to reset the epigenome and treat ovarian cancer. So with that, I'd like to thank you again for being here tonight, and I'll hand it over to my colleague, Ron Evans.
Thank you, Diana, and welcome everybody, and it's an honor to be here to help launch this initiative. I came to Salk 39 years ago, and I'm known for work on genetic switches that control metabolism and how metabolism controls cancer. Now, cancer is about facing an enemy from within and often not being able to do anything about it. It's frightening, and many feel helpless. It is a very challenging disease to deal with and tough to get a diagnosis. My dad was a physician, and in 1974 he died of a GBM, brain cancer, the year that I got my PhD at UCLA. My baby sister passed away 3 years ago from kidney cancer, which was a major loss for our family. But what set me on my course for cancer research and the work that we do was the loss of my brother at the age of 30, my older brother at the age of 30, to myeloid leukemia, which at the time had no drugs and no therapy, and there was nothing that we could do for him. I tried to figure out at that age new therapies and was buried in the books, but there was no therapy and we lost him. Yet 14 years later, my lab discovered a secret to that disease. And from that, we were able to identify a drug called retinoic acid, a form of vitamin A, that now is able to cure 90% of the people that get that disease. So although my brother was not able to benefit from it, this was a major advance, and in honor of him, it's what really drove that process. And so people make a difference, your loved ones make a difference, and even if they're not here, they still can make a difference. So why did I come to Salk? Salk, to me, is a brain trust. And I came because many of my scientific heroes were here. When I was a graduate student at UCLA, my advisor would drive me down here to meet Renato Lebecco. I remember seeing him on the first time that I visited, and he was like the godfather, literally. And I sat down in front of his desk and goes, "So Ron, tell me what you do." And that's like, okay, this is it, my life's over. So, but it's an inspiration to be able to meet someone at that early stage of your career who is an amazing person and a huge impact on the field. This was before he won the Nobel Prize, but he was just such a major figure. And at Salk, our daily job is to come and try to tackle the most difficult problems. I'm now working on pancreatic cancer. I'm going to show you a little bit of that in this picture where the cancer is being surrounded by this barrier, and it's generally considered untreatable. And actually, this is not a real image of the cancer or the barrier around it, but rather this is the actual barrier reef, a picture that I took from a plane. But it illustrates the idea. But why does this happen? It really literally gets a shield. Like in Harry Potter, there's the invisibility shield, and the drugs and the immune cells cannot get to it. They can't find it. Our goal is to take down that shield and break up the barrier. So in the end, we believe that knowledge is power. Knowledge really is power. And the way in which we attack this is by identifying a weak spot using genomic sequencing to find that the vitamin D receptor was the key secret to taking apart this barrier. Vitamin D, the sunshine hormone, can unblock that field and unleash the power of the immune system to come in and take out the tumor. And I'm going to just show you a little illustration of that in this little animation where the tumor is locked inside. The vitamin D form that we have comes in, decodes the barrier, and exposes the tumor so that immune cells can now come in. You can see now they are beginning to take out the tumor. This is actually a major conceptual advance, but because it's striking, it has a lot of clinical implications. And so while being a basic scientist, by partnering with our clinical collaborators, not just at UCSD, which we do, but at Memorial Sloan Kettering, at Harvard, at UPenn, in Cambridge, England, we have partners everywhere. Why? Because if you discover something, people want to get it into the clinic. And so we have multiple clinical trials that are ongoing. And with this, we think that this kind of therapy makes something possible that was once thought to be impossible. So I believe there is a treatment and possibly a cure for every cancer. In the end, finally, I just want to say that not only thanks to everybody here, but everyone here can make a difference. Each one of you can help. While Jonas Salk said that hope lies in dreams, you saw that when Ruben had his slides, we in the Cancer Center know that science can turn those dreams into reality. So that by believing in Salk, you believe in the future and possibly in a world without cancer. Thank you.
Okay, very good. One of the questions that a number of our guests asked when they sent in their questions was, is a cure possible in our lifetime? Jeff, you want to start us off on that one?
Sure, thanks, Marna. So this is a common question that's asked. Is there going to be one cure for cancer? You know, if cancer was one disease, I believe we would have found it already. But I think as we've hinted at already, cancer is an umbrella term that describes numerous diseases. And as you've already heard, for some cancers that were thought of as death sentences even 15 years ago, such as the one that Dan Lewis has, there are now treatments to successfully counteract that cancer. Dan is living proof of that, as are many others. Now, there will be cancers that are so complicated that one drug won't be sufficient. And so combinations of therapies will be needed. But even then, probably for a number of cancers, they won't be cured. They may be controlled. Now, I think that a person who could live with instead of die from their cancer would be happy with such control. But I think we've only touched the tip of the iceberg. We're only starting to mine the depth of the knowledge that we're gaining. And we can use that to fuel the development of other approaches, such as I think Sue might want to talk about.
Yeah. So as I mentioned, immunotherapy is revolutionizing cancer care. And probably every treatment going forward will involve immunotherapy with another combination. And we are seeing cures today. There are a fraction of patients they have metastatic cancer, they're going to die in a few months, they get this immunotherapy treatment, and their tumors melt away. Metastatic disease evaporates, and they've been living now for many years. But the reality is that this is a fraction of patients, and so what we have to figure out is how can we make the numbers of patients benefit? How can we combine these treatments? Understand we got to incorporate many different processes that are occurring within the tumor to cause the tumor to grow, to evade the immune system, to change the other cell types that are in that environment that are allowing that tumor to progress, because they're all feeding that tumor growth. So we need to intervene at multiple different ways, and that is only now just starting. So yes, I'm very optimistic that we will be curing much larger numbers of patients in our lifetime.
We've talked a lot about cures, but what about the risk factors? Diana, what are the risk factors for cancer?
Yeah, this is an important question. Understanding risk is very important. We know that the top risk factors are heredity, age, and environment. So cancer is fundamentally a disease that is caused by mutations in our genes. So anything that accelerates the number of mutations in our genes will be carcinogenic. And there's good examples of this like UV exposure or smoking. But it's also true that heredity is a big factor. And this is because many people are born with one of the two copies of genes already damaged at birth. These people have a much greater risk because they only need one hit for cancer to develop. And so this is for sure a huge risk factor. But we know that there is also other risk factors among healthy individuals, non-smokers. Cancer is still a huge problem. And so we're just beginning to understand how maybe lifestyle is impacting cancer risk. And I think Ron has done a lot of work in this area and can maybe speak to the contribution of lifestyles.
Yeah, thank you very much. And those are all really good points. Risk is a key factor here. I like to think of it in three parts. One is knowing your enemy, and what your enemy might be, how it might be lurking. Know yourself, and then take control. And the enemy, in a sense, partly it's the heredity, partly it's risk factors in the environment. Stress is an enemy, which we all experience. High-fat diets,
Like
obesity,
right
sedentary behavior. I mean, and I'm sorry, this is like a depressing talk. But the environment actually has an impact on us, and cigarettes, you heard, all these things that are somewhat voluntary and some are involuntary. Pollutants are not voluntary, but we're exposed to them. And so these do have an impact, so you need to keep that in mind. It's important in terms of taking control to make sure that you go to physicians, get checkups, pay attention to what they're saying. Know if you have a risk, to be sure to stay on top of that. And also humans are very, very good at moving. Exercise is a known factor that reduces risk for just about every form of cancer. And you don't have to be a marathon runner to get the benefit. For exercise, mild exercise gives you most of the benefits. So a little bit of exercise goes a long way. The last part that relates to aging, this is important. I will sort of call out my elderly colleague, Jeff Wall, to talk about that.
He can,
Did you hear me?
yeah. He can only do that because our birthdays are about 2 weeks apart. And my last one put me into a slightly different decade than did his. So he's catching up to me rapidly.
I'm in my 60s, he's in his 70s.
Yeah.
Big difference.
Right. So
You're doing something right.
I'm exercising. I'm exercising. Keeping a low-fat diet, you know. Ron and I always look at the amount of vegetables that we have on our plates. You know, this is very important. But aging is very important, and because of good medical care, of course, we're all living longer. Why is aging a factor? Well, as Diana indicated, cancer is a problem where you have to, one mutation doesn't cause the cancer, one change in the DNA doesn't cause the cancer, but many changes in the DNA are required to cause the cancer. But you know what? That isn't enough. I'll tell you how I know that isn't enough. Because a study was published recently in which they looked at the only sun-exposed part of your body that's always exposed to the sun. That's in your eyelids.
you were
And,
gonna
you
say on
know,
top of your
very few. Can wear a hat, can wear a hat, excuse me. Anyway, for the great majority of people, maybe in other audiences, it's the sun exposed, it's the eyelids. And so people say, well, why don't people get cancers in their eyelids? Maybe the eyelids, maybe the cells of the eyelids don't accumulate mutations. Turns out that's not true. The cells in the eyelids accumulate mutations at the same rate as cells in other places do. But there's something else that's different. The structure of the eyelids is maintained. Some way, our body has a way of really renewing those cells and maintaining the structure. So cancer can be contained by having a normal structure around it. It's when that structure becomes abnormal and permissive that the cancer is allowed to grow. And in fact, in pancreatic cancer that Ron talked about, it's amazing to me. Ron got me involved in pancreatic cancer, so thank you, Ron. Only 10% of the cells in the pancreatic cancer are actually pancreatic cancer cells. Those cells are kind of massaging or manicuring the rest of their microenvironment, the rest of the environment surrounding them to enable the cancer cells to survive and thrive. So with aging, we have changes in our body, decreases in the effectiveness of the immune system, consistent or persistent inflammatory situations that arise that change the structures that enable cells that are already initiated to progress.
Right.
I remembered that, Ron. That's great.
Ruben, do you want to say something about metabolism here?
Yeah, well, to echo and to follow up on what Ron said, I think we also have only in the past 5 years or so really learned some of the pathways that exercise impinges on, and also diet impinges on them. So actually by eating a good diet, believe it or not, you by keeping balance in your metabolism, you actually also keep balance in some of these growth control points that effectively keep the cancer in check. And we haven't historically thought about things that way, that exercise or diet could actually be regulating enzymes in all the different cells of our body, but incredibly they are. So that's exciting, and I think ties in nicely with different things that people are working on here. So
Great.
Stay tuned.
Thank you. Jeff, you made the point that there isn't one cancer. There are many cancers. What do the cancers have in common? What do you look for?
So I think that there are two really important parts of that question, Marna. One is, what are the underlying features of cancer? And the second one that's really important for treatment is how do we identify the commonalities so that we can use them as a strength for us to develop drugs that are targeted against commonalities that we can use in different cancers. Okay, so what are the commonalities? Well, we've already talked about some of them. Cancers all have to have mutations in genes that control their ability to grow, to survive, to evade drugs, to gain this plasticity that I talked about, to become stem-like, and to move and colonize different sites. Within this, there are enzymes such as the one that Tony discovered and now is the founding member of 530. How many of them are there now, Tony? 35, 52, 550 something?
540, right? So
See, even Tony can remember, okay. But Tony has an incredible memory. So 540 of these kind of kinases, tyrosine kinases. Knowing that there are all these kinases that are related to one another that can drive different cancers, we can start to target them and see whether the drugs used to target one can be effective in another. So let me give another example with Dan. So the enzyme that drives chronic myelogenous leukemia is inhibited by the drug Gleevec. Turns out that drug was developed for another cancer, for another kind of enzyme. That kind of enzyme is mutated in a cancer of the GI tract. So Gleevec can not only be used for CML, it can be used for a cancer of the GI tract. So by thinking about cancers not as diseases of organs, but as diseases of genes and of enzymes and processes. Finding out the processes that are in common can empower us to find drugs that can target many types of cancer that we normally wouldn't have treated in that way. There's another commonality that Sue may want to talk about regarding the immune system.
Yeah, I think that an underlying feature of all tumors is the non-tumor cells that Jeff was just talking about. All tumors are surrounded by cells of our immune system, like macrophages, by stromal cells, fibroblasts. These are all common features, and they get manipulated by the cancer. And so a promise of immunotherapy in particular is that, and strategies that target these non-cancer cells, that there'll be more common principles that underlie their function and the way that they're being manipulated in cancer, that drugs aimed at those cells, so not the cancer cells at all, aiming drugs at the non-cancer cells may hold promise for treating a larger number of cancers, because those might have more common principles across different cancer types and different types of organs. And I think Diana wants to speak about
I mean, this is absolutely the hope for epigenetic therapy as well, because we know that epigenetic processes are almost universally dysregulated in cancer. And so the hope really is that we can find those common nodes, the commonality between cancers, again, independent of origin, and that by hitting those nodes, we could really develop drugs that would be effective in a broad range of cancers.
But I think I wanted to also say that there are so many differences with different types of cancers as well. And again, thinking about our immune system, one of the biggest differences is going to be each of these different mutations, every tumor and every person's tumor, and even when you think more broadly about within a single patient and their metastatic cancers, each of those tumors have different mutational profiles. Some are in common, but some are different for each of those tumors. And so those are now presenting themselves as distinct features, flags on the tumor cells that our immune system can recognize. So while there is going to be some commonality in the treatments we use for cancer, there are also a lot of strategies being aimed for personalized medicine to stimulate the T cells to attack the actual mutations in your tumor that are causing these flags looking like non-self. And so there's going to be uncommon, very personalized approaches as well for different therapies. So.
Diana, we've talked a lot, and we heard Ruben say that Salk was a very special place, well suited to the cancer research and finding cures. What's unique about Salk? How do you find it a very special place for supporting your cancer research?
Yeah, I mean, absolutely. I think as sort of the most junior person here, I've been kind of welcomed with open arms, and it really is this collaborative, collegial environment that really speaks to team science. I think this was the vision of Jonas Salk, to bring the best and the brightest, kind of the French Salon of La Jolla, California, and to really use sort of new ways of thinking about old problems. And still to this day, I think our activities, our departments, our architecture is still there in place to facilitate interactions that would normally not occur in traditional building environments or department environments. And for my own example, I am very focused on the molecular mechanisms. I have my head buried in the DNA. But so I don't typically think about the cell or the environment at all. But I think when
We'll help you.
Yeah, absolutely. So when I found that my cells were starting to produce things that are known to attract immune cells, it was easy for me to knock on Ron's door, grab a coffee with Sue, and now my lab has really started to initiate a focus on the cross-section of epigenetics, the immune system, and immuno-oncology, which has been very exciting for us. And I think, at other places, this really would have not been encouraged, but when I joined the Salk, Tony said, we expect you to be doing something different in 5 years. And I see that that's already happening in year 2.5. So,
and Diana's a trained immunologist.
Right, yeah, coming back to my roots, finally. But yeah, so I believe it. It's not something that was, everything that was told to me, I'm finding true.
The secret sauce
The
exists.
secret sauce is, yeah.
Wonderful. Ron, Ruben talked about speeding up the process from bench to bedside to bench. And we are a cancer center, but we don't have patients. We're not a medical center. How can Salk do that? What's the process for speeding up that, for getting more involved with clinical trials?
I like the term speed because I like race cars. So does Jeff. And
I don't.
I
like my
But you know, the thing about speed is that it takes power, but highly controlled. And so Salk is an environment where ideas really drive powerful discovery. But it's not enough just to have the discovery. You have to be able to move that into a place where you can work with clinical collaborators. But what I've found that is the case is that if we make a discovery here at Salk, everyone wants to work with us. They're not going to be made at some of the clinical centers. The best clinical centers have the best capacity to turn these into therapeutics. And so they're so happy. You don't have to be big to have a big impact. You have to have a big idea, and that can come and does come from Salk. And so that's our trick. And the clinical centers love to work with us, number one. And if the discovery is right, you get the big pharma like Merck. We have 2 separate clinical trials with Merck, with one of their immuno-oncology drugs, where our drug, the vitamin D component, unlocks the ability of that immune system to come in. So we can accelerate because we start with our power, but everyone wants to work with us. And if we create those connections correctly, we are the drivers of that entire process, and it's working very well now.
Great. Tony, we saw in the movie about your seminal discovery and many cures that led to. Where are the new opportunities today?
Well, I would say from my perspective, the new opportunities involve developing new tyrosine kinase inhibitors, of course, and a new tyrosine kinase inhibitor was approved by the FDA on Tuesday this week, bringing the number up to 32. So there'll clearly be more progress there. And one of the challenges is that tumors often become resistant to a single agent tyrosine kinase inhibitor, and so one needs to build 2nd and 3rd generation molecules inhibitors that can act on the altered kinase targets. But I think going forward, one of the most important things for us to do is to learn how to use targeted drugs like tyrosine kinase inhibitors in combination with other drugs like epigenetic inhibitors, and also particularly, as Sue was saying, with immune checkpoint inhibitor antibodies. So we need to learn how to rev up the immune system, target the tumor cells, and try and eliminate tumors. Another very important area that we are trying to do something on is to improve early detection of cancers. The reason why pancreatic cancer, glioblastomas, and ovarian cancers are so deadly is because they are always diagnosed too late. And if we had a better way of identifying these tumors early on, for instance through a blood test, perhaps circulating DNA molecules, then I think we would do a much better job of treating these cancers, often surgically. So the other major challenge of cancer is metastasis. How do we identify cells in the tumor itself where not all cells are the same? Tumor cells are very heterogeneous. We need to be able to find the cells that are going to go on and wander around the body and establish a new tumor at a new site. And we need to be able to do that in a much more thoughtful and analytical way. So overall, I'm optimistic that we can use all of our new knowledge to develop better diagnostics for early detection and therapies, and ultimately conquer cancer by turning it into a chronic disease. So it's a disease one dies with rather than of, And I'm absolutely convinced that the Salk cancer scientists, with their innovative and team approach, will be making major contributions in all of those areas.
Thank you, Tony. Unfortunately, we're running out of time, and we have lots more questions from you all, but I hope you see that we have lots of answers, too. So, well, yeah, I'm just going to do that. Thank you. No, keep me on track. As we wrap up, Ruben, you made the point in your presentation that we could be helpful. How would you like us to engage with you to be helpful in conquering cancer?
Yeah, that's great. I just want to, personally thank everyone for being here today. Salk was the first NCI-designated cancer center in San Diego, and this is actually the first time that we have launched an initiative devoted to cancer research in these years. So this is a big dedication and step forward. This is really just the first event. This is literally the first event of things that will go on for the next year and years. And so we're asking you to partner with us, to work with us, and to come with us on this journey as we set out to conquer cancer. It's time now to write the future. Thank you.
Thank you very much again to our panel. I hope from this just short session, you all get a glimpse of why I believe that Salk scientists are going to lead the way to cures in our lifetime.